So this morning I have the pleasure of introducing myself for the first talk. So I'm Veronica Hood. I'm the scientific director. I think you guys have all seen me up here all throughout the week, so you probably know me. But I'm also so pleased to be joined by Dr. Joe Sullivan from UCSF. We are going to present together and give an overview of sort of the concepts of genetic therapies and try to go pretty broad and hopefully have a lot of time for questions. You can ask us anything. I think sometimes it's scary because these topics are so complicated, but we're going to try to make it as simple as we can, and we hope that you'll feel comfortable asking any questions that you have. So what does it mean when we say we want to treat the cause of Dravet syndrome, when we say we want to cure for Dravet syndrome? As we've heard throughout the week, we know that the majority of cases of Dravet syndrome are caused by mutations in the SCN1A gene. And so we think if we can address the dysfunction in the SCN1A gene, that loss of half of that SCN1A, maybe that could do more than just treat seizures. Maybe that could really modify the entire disease. Okay, what does that all really mean when we get into the nitty-gritty? And so what I'm going to do is give you a crash course in genetics and molecular biology and set you up with the background for then Joe to go into the approaches to genetic therapies. So I will put a big asterisk by this. It took me until my master's to really wrap my head around some of the things I'm about to tell you, so I know it's complicated. You know, in my undergrad degree, I could probably recite back to you the things I'm going to tell you, but it probably took me 6 years of biology, college education to really understand it, so I recognize these are really complex topics. I'm going to try to start slow, and so try to just get, a tidbit, and then if you want to go rewatch, and then you'll get the next slide. I'm going to try to build on it, but just know it's complicated, and I know it's complicated, and it's okay if you don't get it the first time around. So we're going to start at the beginning. DNA leads all the genes in your DNA make a protein. So here you've probably seen that double helix of DNA. Contains over 2,000 genes, and each of those genes code for a protein. I like to use this analogy that DNA is like this giant recipe book. And it has lots of recipes, and each recipe is the equivalent of the gene. And from each of those recipes, you can make your meal, which is the protein. So it's the dish. But there's this middle step called RNA. And so when you're thinking about that, so you've got this precious recipe book that's been handed down through your family, and somebody wants to do a recipe, you're probably not going to hand them a page out of the book. You're going to want to protect that original book. You might copy it down onto a recipe card, and then that's what you're going to have on the counter as you're cooking, or that's what you're going to hand to your friend who wants the recipe. So the DNA is really important. That's your master copy of all the genes. So that stays in a really protected part of the cell. When you want to express a gene, when you want to make the protein from the gene, there's the middle step called RNA. And so it's like a recipe card that your cell copies down, and then it can read that recipe card several times and make several of the meals or the protein. All right, and so then if we zoom in even farther on the DNA, lots of you have seen your genetic reports and you've heard us talk about mutations, but what does that really mean? So your DNA, if we unwind those strands and zoom really close in, you'll see that there's represented by these 4 different letters, A, T, C, G. They're called nucleic acids, but that's the genetic code, are these A, T, C, G, and the way that they are organized. Sections of the DNA then are made into that strand of RNA. So you're copying one letter at a time into that RNA strand, so onto your recipe card. Every 3 of those letters that are on your recipe card are like a cipher code for the cell to then make protein. So here you see UAC makes the Y. UCA makes the S. And so this is how your cell is making the protein from the gene. It's reading it in these codes of 3 and then assembling a protein from that. Proteins then get folded up and processed so that they can go do their job in the cell. So what happens when that code gets disrupted? So a mutation causes a change to the DNA code that affects the gene expression or function. Sorry, it should have an animation. There we go. Okay. So here we've replaced one of the letters in the code. That disrupts that code from making the S that you need in that spot. And so you may have heard of truncating mutations or premature stop. You're trucking along. You're supposed to read that whole recipe, but suddenly your recipe says, actually, after step 2, stop. And so now, you're not going to have a very good recipe if you only do the first 2 steps and then put that on the table. And so that's what happens when you have a nonsense mutation, premature stop codon, truncating mutation. Those are all the same thing, and that's what it means. You may have also heard of missense mutations. So this, again, is just changing one letter of that code, but instead of saying stop, it changes that step. And so now you've replaced that step with something very different. And again, that can affect how that protein product works. There are lots of other types of mutations. You can have an insertion, so it's very important. It's reading it in segments of 3. If you add an extra letter in, everything shifts. Then the code is messed up and all of your steps are wrong. Or you might delete a big section of the gene, and then you're missing some of those steps. All right, so if we now take all of this that I've just told you and think about it in the context of Dravet syndrome and the SCN1A gene. So here I've represented in the DNA, this big block is just saying, here's the section of the DNA where the SCN1A gene would be found. That makes mRNA for the SCN1A gene, and then we make the NAV1.1 sodium channel. Everyone should have 2 copies of the SCN1A gene, and you need those 2 copies to make all the sodium channels that you need. But when you have one of those mutations that I just talked about, you lose half of those, and now you only have half the amount of sodium channels that you need. That's called a haploinsufficiency. So the goal with genetic-based therapies would be to fix that haploinsufficiency. There's a lot of other layers to the gene besides just the code that you need for the protein. There's all these layers of gene regulation. And so if I'm going back to that recipe analogy, if you've ever bought like one of those celebrity cookbooks, or if you've gone to a recipe blog to try to just find the recipe, and they've written paragraphs and paragraphs to tell you, I love to make this recipe on the 4th of July, and sometimes I sub out this ingredient for that. Your DNA has all this extra information around the gene that helps your cells know when to turn the gene on and off, how much to express the gene. There's parts in the middle that get edited out so that you make the mRNA transcript. So here I'm showing you at the top the gene. There's a regulatory region often in the front, so some genetic therapies might try to target that to say, turn this on, turn it off, turn it up, turn it down. There, like I said, you have the big section of DNA, but when you're sort of copying down that recipe into RNA, you may not just copy a straight section. You might have little segments along the DNA, and your cell has to go in and cut out the parts that they don't need for the core recipe. Those parts might still have information about when to use the gene, when not, but you don't need it when you go to the RNA. But all of this could be important for targeting genetic therapies even to those regions. So then you just take the important parts and put it into the RNA, and you've really reduced the amount. So the big gene, if you look at the DNA, how long it goes, you have 160,000 letters to make up the SCN1A gene. But when you edit that down to the RNA, it's about 6,000 of those letters. and then you can make the protein from that edited down recipe. Now I'm gonna pass it off to Joe, now that I've given you all that background, to talk about how scientists are getting creative and targeting all those areas of the gene to hopefully fix the haploinsufficiency.
Thanks, Veronica. You could have been my college genetics biology teacher, and I understood it a lot better from you, so it's great. So now that we have all this alphabet soup, and I love the recipe analogy, I'm a big cook, but I don't like to bake. And I don't like to bake because you have to follow the recipe exactly with baking, right? Whereas if you're just cooking dinner, you can throw a little bit of this, a little bit of that. Whereas baking, we all know that if you're just off by half a teaspoon of something that you added, the product, your cake doesn't rise, your cookies look flat. So we may even be more specific and say that these recipes really are like a baking analogy. So with that sort of alphabet soup, and then you heard, yesterday from Ingo about the C.G.P. And then you heard from the Stoke folks on Thursday about some of their preliminary data. I'm going to try to go through at least the 2 gene therapy trials where are further along, and many of you are probably expressing interest in those trials, so that we actually have an understanding of how we even got to today, where we're actually be able to offer this to human beings, right? Because these are first, like much different than drug trials like Scott Perry talked to you about yesterday. These gene therapy trials are not tried in adult healthy volunteers, right? Like Fintepla was, or like Epidiolex was. Rather, we're going into the disease state that is Dravet syndrome first. So our goals, right? I'm preaching to the choir here. It's not just about seizures, right? We're trying to treat the underlying cause to address the whole host of comorbidities. It doesn't necessarily mean that you need to fix the mutation. Okay, there's a lot of buzz out there with some of these recently approved CRISPR technologies for sickle cell that were just approved late in 2023. And I'm going to show you that those actually don't fix the actual cause, but they actually do some of these workarounds to get at what the underlying dysfunctional protein is, or to have some compensatory mechanism to account for that dysfunctional protein. And even another neurological disorder, spinal muscular atrophy, that actually doesn't, some of those don't even actually fix the SMA1 gene. Rather, they take advantage of this other gene, SMN2, to try and make that work harder. So in that faucet analogy that Kimberly Parkinson gave from Stoke, as long as we can turn up the faucet a little bit to get more of the protein, or at least a protein that can make up for the dysfunctional protein, we still may have an opportunity to address that underlying cause. So that's all background to say, there are multiple strategies to try and turn on this faucet. And so as Veronica gave you this nice sort of backdrop about the DNA, the messenger RNA, and the protein, there's lots of different ways to perturb that pathway in an advantageous way. And the 2 that I'll talk about, because these are the 2 that are farthest along, are 1, to increase. Is that me? Okay. To increase the amount of messenger RNA that actually is productive, right? And then the second one is to actually increase the transcription, but at the DNA level, right? And so these are 2 different ways, but in the end, it's all getting back to increasing the amount of functional sodium channel protein. And then lastly, because I'm sure there's so many questions about CRISPR, and I am not a geneticist, the genetics that I learned in medical school was probably what Veronica just gave you, and obviously there's been a lot of progress since I was in medical school, and I don't like to consider myself that old, but I'm going to go over a little bit of the CRISPR technology to understand what's exciting about it, and not to be the Debbie Downer, but some of the limitations, at least in 2024, that doesn't make it ready for prime time, at least for Dravet syndrome, but things are happening very, very quickly. So you've already seen this little cartoon, both from Scott Perry and from the Stoke group. So this is the non-productive messenger RNA approach. And this is also something that I don't think was known when I was in medical school, right? There's this poison exon that Ingo mentioned the other day. So like this poison exon, why does this exist? It's almost like the appendix, right? There's all these redundancies in our body. Like, why does the appendix exist? It doesn't have a purpose. But they realize that this poison exon is just another way to regulate, like Veronica said, how these genes are actually spliced together to ultimately make the protein. So it's another way for our cells to finally regulate. Whenever I hear these gene talks, I'm amazed, like, how are we even like here. There's like millions of base pairs, and you need, all these things that are happening in dividing cells. Like, how is it that we have even survived as a species? It's pretty amazing to me that more things actually don't go wrong. So this non-productive mRNA approach, again, is focusing on the fact that there are these alternative splicing events, okay? And so what we're seeing here in the blue is this sort of poison exon that is sometimes included in that recipe, okay? So imagine reading the recipe and then it tells you to stop after you add your half cup sugar. That recipe is not going to come out well, and that's kind of shown here where you get the no protein, no cake rises in the oven, right? And this is actually happening in the pathogenic variant of the SCN1A gene that leads to Dravet syndrome as well, but those basically has the same net result, right? The missense mutation or the nonsense mutation, you don't get that productive protein, and you don't get it even when that poison exon is included. So this approach, this TANGO approach, which stands for Targeted Augmented Nuclear Gene Output, right? I love it when acronyms actually make sense because it actually tells you what it's doing. So it's targeting a gene to increase its genetic output. And it's almost like a sort of, do not disturb sign is the way I kind of, so it's this ASO binds to basically exclude this poison exon, right? So now this poison exon is removed and allows the rest of the recipe to actually be read. And the idea is if that is actually effective, that will actually translate into more of this functional sodium channel protein. Again, it still happens in the mutated gene as well, but because that extra messenger RNA exclusion is still translated, is still read because of the variant that leads to the loss of function, it doesn't actually have any positive or negative effect. And so this was the approach. And you saw from the Stoke presentation on Thursday how this is actually translated, at least in the phase 1/2 clinical trial, into improvements in seizures and starting to see some signals about how this is improving some of the non-seizure related comorbidities. But how did we get there? Like, how did we actually go from this theoretical approach to say, okay, this is actually safe to give to a child? And so this is where we start with the lovely little mice, right? So mouse models, I think, we've talked a lot about how far ahead in many ways we are in SCN1A related epilepsy disorders because we've understood the gene for a long period of time compared to some of the other more recently discovered genes, which we're trying to understand. The fact that we have an animal model really allows us to try a lot of different things in a controlled scenario to understand if they are ready to move it to the next step. And so this may all sound good, but actually, does it actually do that? Because keep in mind, for our patients, our kids, our loved ones, we actually can't, thankfully, grind up their brains and look at, is the messenger RNA increasing? Is the sodium channel increasing? And we can't measure these things, right? So all we really are left with is measuring, are they improving? Are seizures improving? And that's kind of a surrogate marker to say, okay, we think what was shown in the mouse is actually also happening in the human. And so this was a study that was done in a little baby mouse where they're given at the postnatal day, these numbers P2 means second day of life. They gave this ASO, right? So something very similar to the Stoke ASO. And then 7 days later, they can actually sacrifice the mouse and grind up the brain and look up lots of these different proteins. And so what you can see here in this first figure, and this is the dose. So on the x-axis here, they're giving increasing doses, right? If you have all of this extra poison exon that's circulating around, you want to make sure that you take full advantage and exclude it as much as possible. And so you may need to give more of the ASO to target all of the poison exon to sort of turn that faucet up as much as possible. And so this actually shows that non-productive transcript, right, the one that includes the poison exon, as you actually increase the dose of the ASO, the amount of that non-productive exon goes down. And you can kind of see that once you sort of have them all labeled, right, once you're excluding all those exons, giving higher doses doesn't actually translate into any more benefit, right? So that's helpful for us as well because every treatment that we think about in medicine has risks, benefits, side effects, right? And so if we can try to arrive at the minimal dose effective just suppress this non-productive and then translate into the productive. Like you can almost do a mirror image here. So this is exactly what we would anticipate would happen. If you decrease the non-productive transcript, you're going to increase the productive transcript, but then it still needs to translate into the cake being made, right? And so that's what's actually being shown here in terms of the functional NAV 1.1, the sodium channel protein. So this was, in many ways in science, I don't think you could actually have hoped for any better results here in terms of going into sort of a proof of principle as to whether or not this is effective. And this is a busy slide, but it kind of goes through it in even more detail and kind of to highlight, okay, how long does this effect last, right? We may actually see, I showed you back here, this is the mouse at 7 days, but we don't want to have to give this type of medication through a spinal tap, a lumbar puncture every 7 days, right? We want to make sure that the treatment effects are actually longer lasting. And so this is looking at these same mice over different time points, 7 weeks and 14 weeks. On the first panel here, C, we can see this is the concentration of the actual ASO, and you can see that it actually sticks around. So 7 weeks, it's still around. 14 weeks, it's still around. And then this PBS, that this is basically, everything needs to be given a control. We actually give placebos to mice as well, right? Because we need to have this be as clean as possible to understand what the natural variation is over time. And so you can see that there's no change here in those placebo-treated mice. And then you extrapolate this out in terms of the percent fold change, or not percent fold, but just fold change in the SCN1A gene expression, and that's that messenger RNA, and then the actual protein. And hopefully you can all see this, but right here is 1, right? So the normal wild type does not have an SCN1A mutation. That's kind of the baseline. That's considered normal, right? So they're at that 1. And then whereas the SCN1A gene, you can actually see that they're actually going to be expected to be less, right? And so when you give the ASO, we can see that that productive, or that the mRNA transcript increases almost 1.5-fold in the actual wild type. So you give it to a healthy person, you give it to us, it will increase the amount of sodium channel. And that, as you heard from Ingo yesterday, there's these gain-of-function mutations. Too much can also be not a good thing, right? So we wanted to make sure, the investigators here wanted to make sure that increasing that too much didn't translate into side effects as well. But then more importantly, in the SCN1A mouse, you can actually see that it went up 2-fold. So trying to restore what we would expect to see compared to someone who does not have one of these pathogenic variants. And that was persistent over 14 weeks. And so that's how the dosing was somewhat informed, and for, at least in terms of trying to do the dosing every 3 months. So that's all fine and good. So yes, you can show that the protein gets increased, but does it actually get increased in a way that's meaningful, right? Does it get inserted into the neuronal membrane? Does it actually have an impact on that neuronal instability and translate first and foremost into improvements in seizures? And thankfully the answer to that is yes. And this is again showing the mouse where they're dosed. Then they actually have a little brain surgery that's done at around 20 days of life, and then they record directly from the mouse's brain so that they can actually see not only behavioral seizures, these mice do actually have convulsive seizures, but we also want to see what impact it is actually having on the background EEG activity, including electrographic seizures. And so you can see it doesn't stop the seizures. I'm sure there's going to be many questions, why does it not stop the seizures? And I'll just warn you, I don't have an answer for that. But hopefully you can see that there's a dramatic reduction, right, in the number of seizures in the treated animals. And then the latency, so the time in which to then they have their actually first seizure is actually also significantly prolonged. And then what about survival, right? Something that is obviously very difficult for us to talk about, but SUDEP is very common in Dravet syndrome, and it's very common in the Dravet mice. In fact, it's often the most common form of death in these mice. They actually don't live very long. And so we can see here that in the treated mice, all but one, and this always scratched everyone's head, why did that one die? And again, we don't have the answer to that, but you can see a nice separation here between the treated animals and those that are untreated in terms of how long they survived after dosing. So that's the sort of ASO Stoke approach. And I know that the Stoke group mentioned, so the phase 1/2 has been completed. Those were the data that they showed to you on Thursday. And the planning is underway for FDA submission of the protocol to hopefully get the phase 3 up and going early next year. All right, so now the AAV approach. This is a completely different approach. Okay, so this is the encoded approach that's the farthest along. And so what is an AAV? So an AAV is an adeno-associated virus, right? So it's a virus that's out there in nature. But interestingly, it actually is not known to cause any disease in humans. It's very small, so it's this 26 nanometer capsid. And I have a little video that I'm going to show after this that'll kind of highlight the beauty of YouTube. You can search these things and find these great little cartoons that can do it much better justice than I can standing up here. And the cool thing is you can basically think about this AAV as like a dump truck, right? It's a transport mechanism to try and get your cargo into the area of the cell where you want it to be, okay? Dump trucks, though, are not unlimited capacity, right? And so one of the downsides as it relates to SCN1A is that you can only fit about 4, like Veronica mentioned the number of bases, 4.7 or 4,700 bases into this cargo parcel, right? Whereas the SCN1A gene, if we're trying to deliver the entire gene, it's just too big, right? There's lots of things that are going on to try and develop better dump trucks, and I'll sort of end with that, but right now the AAV9 approach here, it just the whole gene can't fit in. But the really cool thing is that and this is the part where I will not be able to explain it to you in terms of these molecular biology techniques, of how they can remove the viral DNA completely, and you still keep the dump truck sort of analogy, and replace it with any cargo, right? So they can put in these different gene cassettes and then deliver this virus to a mouse, a human, to deliver that to the targeted site. So here's the cool little video, and I'm just going to be quiet for a second. Is the sound not working? Oh wait, this is when it doesn't have the sound. This is when I have to narrate, sorry. So here's the cool little, why is it? Can we click on that? I might have, there we go, thank you. So here's the adeno-associated virus. Okay, it's going to bind to the cell membrane and go inside the cell. And now it's this thing called the endosome. So again, think of this dump truck analogy, and this is the cargo, okay? So, and then this is the DNA strand that is represented as that cargo. And this may actually be the area of interest. So now this is freely floating inside the cell, and then if you remember back to your high school biology, then there's the nucleus in the center of the cell where all the DNA is. So now this cool little capsid gets dissolved by the cell, then this endosome fuses with the nuclear membrane and releases the cargo. Dump truck opens up. And then in a very cool way, it forms this stable circular, what's called an structure called an episome, which then remains stable. So this is not integrating into the SCN1A gene. It's not integrating into so it can be passed on to future generations, but rather it's still sitting there and still exposed to all of the normal cellular machinery that actually can replicate and, like Veronica said, make more protein, right? So it's a way to get cargo into the cell and then have it be continually made because it just lives in there in a stable manner, okay? And so this is kind of the approach that the encoded team has taken on with this AAV approach. And so these are the cartoons that are taken directly from their website. So similar to that animation, we have this AAV. It binds with the cell, and then it goes into the neuron. This is meant to be a neuron, and then fuses with the nucleus and releases its cargo, so the dump truck here. And now we have this stable episome. And what this episome is, right, is actually a sequence that can bind upstream of the SCN1A gene and basically tell it to turn the faucet on, make more, right? I love that analogy that Kim gave on Thursday. And so after it enters into the cell, the normal cellular, our own cells, the own mechanisms, start to recognize this episome as DNA and read it, right? And then it makes this transcription factor. And then what happens is this transcription factor goes and it binds. So this is meant to depict the normal SCN1A gene. It binds upstream from it, right? So at the start of the recipe, estimated duration, hands-on duration, 4 hours. It binds there. And so once it binds there, it is actually able to recruit in what are called these transcription, these promoters, right? So it brings in more of these reading frames to allow the normal copy of the SCN1A gene to actually be read out more, right? So producing more, again, that faucet analogy. And as far as we know, based on the animal models, this episome remains stable in there, right? This is one advantage and disadvantage to neurons, right? Neurons don't divide. When we're born, we have as many neurons as we're ever going to have, and from day one, we start losing neurons, right? So this episome can actually remain in the neuron, and because the neuron doesn't divide, the idea is that it could be a lifelong potential treatment. So then the idea is that transcription factor, it binds, and then it produces more of the messenger RNA transcript, which then is translated into that sodium channel protein, and and then that's actually inserted into the cellular membrane, and ideally restores that sort of haploinsufficiency. And one thing that I failed to mention is there's actually this other engineered part of this transgene that is really trying to get this cargo to the inhibitory interneurons. So these are the neurons that we feel play the most fundamental role in the underlying pathology of Dravet syndrome, because we don't necessarily want to disrupt other cells if we don't need to, okay? So in the same spirit, does it actually do that? It all sounds great, right? The cartoon looks cool and it looks everything, but does it actually do that? And the answer is thankfully yes. So this was, again, a mouse model that was given the same exact AAV. And we can see here on the top those cells that were not infected with the virus, right? So this is another very detailed that I'm not going to be able to explain to you. I tried to in researching for this talk to learn, and I learned a lot, but to be able to explain, how these assays are done, just take my word for it. So as expected, right, in those SCN1A animals that have the haploinsufficiency, when you look at the cells and look at the number of sodium channels, it's half of what would be expected, right? That's the median, right? There's going to be some that are a little bit different. And then in those animals that were dosed with this AAV, you can see that there's a shifting to the right, not necessarily to 1, right, the median expression, but it's, 0.75 or so in those that were infected. And specifically, these are these inhibitory GABAergic interneurons. So really proof, as much proof as we can possibly have that the intended consequence is actually delivered. And so then when we look in the same way at how those actual mice do, postnatal day 1 and then 28 days later, they're actually sacrificed, we can actually see here the normal mice that do not have an SCN1A gene mutation. You can see there's scatter here, right? But it's meant to be normed around 100%, being normal. Then those that actually have the SCN1A mutation but are just given placebo, right? We can see it's about 50%, again, with a little bit of scatter. That's what these little whisker error bars are meant to show here. But then when they're given the AAV, there's clear upregulation. Doesn't necessarily, the median doesn't, at least in this mouse model, at this dose, does not actually go back to normal, but it's going in that direction, and you can see these p-values from a statistical standpoint are highly, highly, highly significant. And then furthermore, does that actual increase in the sodium channel, is it functional sodium channel, is it inserting into the membrane, and is it actually translating into clinical improvement, which is what we care about the most. And we can see here the answer to that is thankfully, again, yes. That while there still are some seizures in the treated mice, there's a dramatic reduction. And furthermore, we all know our kids are sensitive to fevers and high temperatures. When you put these mice in a box and heat them up, it takes a lot more higher temperatures in order to provoke a seizure. So also trying to recapitulate sort of the human state. And then similar to what I showed you with the Stoke ASO, the impact on survival, these are the treated animals, and this is the untreated animals, so placebo plus placebo, and then these are the actual treated, and we can see that the number of days elapsed is clear separation. Okay, so that's where I would say we are in June 2024. And the ENCODED trial has started enrolling. They have 3 different programs around the globe. One is in Australia, one is in the UK, and one is here in the US. Myself and Scott Perry right now are sites, and there may be other sites that are opening up. And if you have questions afterwards about inclusion, exclusion criteria, that's the hard part. It's not available to everyone. There's ages and there's seizure requirements and whatnot. And I can answer any questions as to why that may be. Okay, so CRISPR, this is like, what about CRISPR? It's in the New York Times, it's everywhere, right? I was very fortunate, the Jennifer Doudna, who won the Nobel Prize for basically exploiting CRISPR in terms of using it in the modern genetic world, is in the Bay Area. I'm from UCSF. I just had the opportunity to hear her speak a couple weeks ago at our resident graduation, and it's just amazing, like how, it's so inspiring to see her stand up on a stage and see that something that she developed is now translating into, disease cures for things like sickle cell. So it's really, really amazing. But I want to also discuss some of the limitations for central nervous system disorders, right? The brain, of course, I'm biased as a neurologist, the brain is the most complex and best organ, right? It's all about the brain. And because of that, there's a lot of protective biological, the blood-brain barrier, right, that protects our brain from toxins and things. But that also is a little bit of a hurdle to get over for some of these therapies. So in contrast to Tango, this doesn't really tell me a whole lot, CRISPR, but I just wanted to put it out there so we know what it stands for. Clustered, regularly interspersed, short palindromic repeats. You can forget that, but that's what it actually means. And it's really cool. This is actually was something that it's a method that bacteria use to fight infection, to fight off viral infections. And essentially what it is this pair of molecular scissors, and I have another little video that I'm going to show you, that allows the DNA of the virus, right, the invading virus, to be cut and therefore be disabled and killed. And so it's their mechanism to fight off disease. But what the Doudna group basically exploited was that could we actually manipulate this system, these precise molecular scissors, to actually cut genes in animals and then ultimately in humans to better understand the disease, right? These mouse models and things can be created with this technology, but hopefully that ultimately translates into some human therapies. Okay, so another video. This one does have a little bit of narration, but it's not playing. Can we try one more time? Is that what? No? All right, I'll narrate then. Let me start over. Okay, so this is a cell. Okay, this is a DNA strand. And this is the sort of Cas9 complex, right? So these are circulating around and looking for a place in the DNA to actually bind, right? And so when this virus actually infects the bacteria, it inserts its DNA into the cell, and it's floating around, and it's hopefully going to replicate to actually infect the cell and set up a host, right? That's what viruses do. They need a host. But this Cas9 system basically recognizes this foreign DNA material, and it creates kind of a mirror image, and then it binds to this protein, which are these molecular scissors, right? So then the next time the virus comes in, it says, hey, I've seen you before. I'm going to bind to you, and I'm going to cut you, right? And so there's these precise molecular scissors that cause this double-stranded break in the DNA, right? And then the DNA, we all have DNA damage. You go out in the sun, you have DNA damage, and our cells actually try to fix this, right? And so the bacteria, the virus DNA that tries to fix itself, but in doing so, it's highly error-prone, right? And so when it's error prone, you get these insertions and deletions, and ultimately it's unable to fix it, and the system, the DNA, doesn't able to bind back together. But what this system with the Cas9 is being exploited now is there are other ways which once it's broken, once the double-strand DNA is broken, is there a way to say, hey, I have this window of opportunity to insert what I want to insert there, right? And that's what really got this CRISPR-Cas9 thing all sort of started, and that's what's meant to be shown here in that video. Okay, but the key here is that cuts are not edits, right? So remember, this was a mechanism for bacteria to disable this invading virus, right? So cutting it allows the DNA. So it actually still has a lot of value in science, right? Because we can knock down genes, we can and understand what a disease actually looks like when you eliminate the gene. But it doesn't necessarily mean in this form that we have the ability to go in there and fix it, right? So the error in the recipe, it doesn't allow us to go in and change a quarter cup to a half a cup, right? And as I mentioned in the cartoon, these two sort of endogenous mechanisms of the cell to repair, are error prone, and although this is more accurate, it also has, some limitations. So what we really care most about, right, the value of having a genetic diagnosis is that maybe we actually know, back to Ingo's C.G.P. Where can we fix, can we actually fix? The problem is that at least, and I looked this morning to make sure I wasn't a couple months behind, that the current base editing systems, right, to convert those C to a G or a T to an A, we don't actually have the tools available yet to make every single switch. And so while sickle cell, right, got all of the press about it is CRISPR-based, but it's actually, in sickle cell, actually is caused by one recurrent gene mutation, right? It's caused by this GAG to GTG change, right? Every patient with sickle cell anemia has that change. In contrast to Dravet syndrome, right, this is probably already outdated. There's probably over 1,800 to 2,000 pathogenic variants, right? So to think of a library that one would have to create on basically a single cargo gene editing sort of package to go in and quote unquote fix or edit every single patient's, I'm not saying it's impossible, but it's just a matter of scale. And so this is actually a figure that's meant to sort of highlight many of the different potential approaches for many of the different genetic epilepsies. Ingo had mentioned yesterday, there's loss of function, there's gain of function, and there's this other type called dominant negative. We're not going to get into all the details there, but let's focus on loss of function. If you can just follow this bold sort of arrow line, if we're really going to use CRISPR, in my opinion, and the opinion of these authors, it really is going to be this sort of promoter epigenome editing. So not actually going in and fixing your loved one's actual C.P.G. Sorry, I'm screwing it up already, Ingo. But actually to bind, right, upstream from the gene to recruit in some of those promoters to, again, turn the faucet on higher. That's not to say that these base editing and prime editing techniques are not going to be potential options in the future, but as of right now, I think the collective opinion is these epigenome editing, right, is probably the direction to go. And this is my last sort of cartoon here to show that there's a lot of cool stuff going on in this field. There's not only to try and get at the different base and prime editors, but the dump trucks, right? How can we get bigger dump trucks that can get across that blood-brain barrier, maybe even be able to be given through an IV? There's a lot of really brilliant people that are working on using different viruses. That's meant to be shown down here. The lentivirus actually has a bigger cargo, so maybe we could get all these components into that dump truck. And then this whole work in these nanoparticles, which again, all you need to do is try to package up the editing or the system that you want to get into the neuron and get it across the blood-brain barrier. And this is just kind of a very busy cartoon to show multiple different ways you can do that. Someone may ask, why not just get 2 dump trucks? It's actually a great idea, right? People are actually looking at that, trying to do 2 separate deliveries, one to maybe do the cut, and then the other one to sort of deliver the cargo, but there's sort of practical limitations that are there as well. So my summary is, Dravet syndrome, our summary, is that SCN1A Dravet syndrome, due to haploinsufficiency, really does allow for a variety of different approaches, right? We don't necessarily have to fix the actual variant. We can exploit that wild type gene that is still there and healthy and working. Right now, CRISPR gene editing may not be the best way to go. I think things are obviously going to evolve very, very rapidly. And again, the real advances are really in these delivery systems to try and just get these really, really powerful tools into the place where we actually want them. So I'm going to turn it back over to Veronica to talk about some things on the horizon.
So I just quickly wanted to reflect on some of the other things that are in earlier stages, just because I think it's really hopeful that it's not just these 2 approaches that are entering the clinic or moving through the clinic. There's so many more exciting ways scientists are trying to target this. And so, if one of these don't come through to be, approved by the FDA, there are many more following behind trying to get there. So there's a lot to be hopeful for. So we talked about the regulatory region, that part that's sort of telling you when, where, how much to use this recipe. And so that's where Encoded is targeting. But that's also, like Joe said, where some of the alternative CRISPR techniques are targeting. So where it's not going in and making a cut in the DNA. We've gotten rid of the scissor part of the CRISPR, but we're just having it go in and where it can recognize the DNA and bind, where you saw in that cartoon. It's then telling the cell, here, let's flag this regulatory region, make more of this gene. Very similar to what Encoded is doing. And there's actually other groups that are looking at other parts of the regulatory region to do something similar to, not with CRISPR, but just like Encoded is doing. There are groups trying to figure out how can we deliver in the dump truck this really big gene that doesn't fit in the AAVs. And so there are people looking at alternative dump trucks, alternative vectors that are big enough to carry it in. And then just like Joe said, there are some groups that are trying to figure out if we send 2 dump trucks in, can we split the gene in half and then reassemble it when it gets back in the cell, which is pretty exciting. And there's just a mouse paper that came out in the last month showing that it was feasible in a mouse. And then there are people that are targeting that RNA. So just like Stoke is sort of going after that RNA step and trying to say, use this iteration of the recipe and not that one. There's also a company called Tavard Biosciences that is targeting that mRNA step through another type of RNA that can bind to it. And so I have a very small cartoon there on the right that sort of explains 2 of the approaches that Tavard is looking at. One of them that can have an RNA come in and sort of cover up if you have a premature stop codon. If your recipe is incorrectly telling you to stop, it can sort of cover up and say, no, ignore that, keep reading. And then there's another approach that, instead of these that bind at the DNA level to say make more, there are some approaches that can bind that mRNA strand and say, be a little more stable, stay around a little longer so we can make more recipe from you. And then, when we're thinking about these targeted therapies, there's actually groups also moving out of the genetic material and looking directly at the protein. And is there a way that we can target the sodium channel to make it more stable, make it last longer, make the ones that we're making more efficient? And so I just think it's so hopeful to see all these different exciting things. And I think this talk is hopefully going to be outdated in a couple years because science is moving so fast, so the things we're telling you that aren't possible right now might be possible. I hope we are wrong on some of these things because where right now CRISPR seems impossible that we could address all those mutations, I think there's the chance in the future that might change. And so we tried to gather some of the frequently asked questions, but I think we threw those up there, but I think we'd like to start with the audience and see what questions do you guys have for Joe and I about this topic. All right, so they're gonna run microphones, so wait till you have the mic so we get it.
It's not on yet. Oh, questions online.
Hello.
Okay, let me
Oh,
do it.
sorry.
Yeah,
And
right.
y'all may have already answered this, but this is way over my head. My son has a nonsense mutation, I guess. So the dump truck or whatever would bring in, like, instead, so his just stops, right? That's my understanding of the nonsense. So it would bring in the whole set instead of what he just has, correct? I'm sorry, I'm trying to clarify. I don't understand all of it.
No, it's a great, are both of our things on? Can you hear us okay? Yeah, so that's the nice thing about we're looking at the healthy copy in both of these approaches, right? We're looking at the healthy copy of the gene. So it actually doesn't matter as long as it's considered loss of function Dravet syndrome, right? It actually doesn't matter what type of mutation one has, because we're still leveraging the normal working copy to sort of turn up that faucet a little bit higher. Do we want to go to we're going to alternate the online question?
We do have a couple of online questions. So you actually answered one of them, but another one was, is there age limitations for gene therapy, and if so, why?
So as far as we know right now, there's not age limitations. I think the general feeling is that earlier is going to be better and more impactful. We don't necessarily feel that we can significantly wind back on the clock. That being said, there are some these mouse studies that have been done that have actually turned on and turned off. That's one cool thing that can be done with the mice, right? They can turn on and off the SCN1A gene at different age points. And in the mouse, in a juvenile mouse, in this one lab that actually showed that it could still have a significant impact and sort of rescue, is the term, the phenotype with regard to seizures and some of the cognitive things. And I mean, I don't need to tell some of you that have older children in the audience, right? Once someone is on a better therapy, even if it's not a gene therapy, regardless if they're 25, 35, we still like to think that they can do better. So.
And it's different than some of the muscular disorders where the cells are dying and you can't target them anymore. For the most part, all those cells that we need to target are still there in the brain, and so there is potential for improvement at any age, I think.
Yeah.
So for obvious reasons, the focus is on the brain, but the SCN1A gene is expressed in other areas of the body, like there's been some talk about heart expression, and that may contribute to SUDEP. Is anyone looking at upregulating in other areas of the body?
Yeah, it's a great question. So certainly the SCN1A gene is expressed in other regions, and it is expressed in the heart. The idea here, though, is once it's in the brain, there's very little off-target. So not only SCN1A outside of the central nervous system, but equally importantly, there's not impact on other sodium channel genes. So I think it remains to be seen. I think there's still a lot of work going on to understand what relationship SCN1A variants in cardiac cells could play, at least into SUDEP. But right now, the focus is CNS.
Yeah, I've heard a couple of times about that some of these approaches may be a one-time only shot, and I'm wondering which ones those would be and why that would be.
So that's right now, that's going to be the ones primarily that are delivered by a viral vector. And one of the reasons for that, sort of the first reason that you couldn't be dosed with another, is that once your body has seen that viral vector, even though it's not actually, delivering any viral DNA, you're not actually getting a viral infection, your body has seen it and your immune system is like, oh, this is foreign and it's not supposed to be here, so I'm going to remember it. And now the next time I see it, I'm going to attack it, because that's how our immune system, keeps us healthy normally. But so you can have a bigger immune response to a second dose of the same viral vector. The other thing that we just have to be careful with is not wanting to boost SCN1A expression too high, right? And so I think there is potential that you could, have, and we've seen it in some other disease states where they got an AAV delivered gene therapy, maybe at a lower dose, they might have saw some benefit, and then later with an approved ASO therapy, which is not delivered via a viral vector, those patients were able to then add some of that therapy on at a little bit lower dose than a patient who was just getting the ASO. This is all theoretical right now. I can't say, yes, that could be the case, for like encoded and Stoke or something, but it is theoretically possible. But we do just have to be so careful that we're not boosting SCN1A expression too high and causing symptoms because of that.
Great, yeah.
The other question that came in online was, does the mutated gene produce mutated NAV 1.1 or any kind of sodium channel? So I think they're just looking for clarification.
No, that's a great question. I think that's this whole loss of function, right? So it really doesn't, assuming you have the clinical spectrum of Dravet syndrome, right? That these mutations, these variants that occur in the gene, it just basically leads, the recipe is not able to be completed, so there is no end product. So maybe my idea of a flattened cake is not a good one. There's no cake, right? Basically, nothing gets made. I think it gets tricky in some of the presenters of, not all SCN1A variants make Dravet syndrome, right? And so that's where it gets a little bit more complicated and interesting. But I think to answer that question, the mutated gene really makes no protein whatsoever, and that's why you end up with this 50% less.
Hi, I just had a question in regards to the medication regime. So after, let's say you went through Encoded's trial or Stoke, right now they're being done while the patients are still on all of their medications. Would you assume that they would need to stay on that regime, or could potentially they pull back just because we know of, the potential side effects of some of these medications?
Yeah, so this is where it gets into the conduct of clinical trials and making sure that the data readout that you're going to get is as clean as possible, right? So one, since we don't know if this is going to work, it is all going to be added on to what we would consider standard of care, right? It would really be unethical to sort of just give someone this and keep our fingers crossed and hope for the best in the absence of any medication. And then furthermore, in order to make sure, is the intervention that we're doing, whatever study it is, this is gene therapy or even medications, it's really important to keep the background medication stable, because if we start going and changing things around and kids get better, kids get worse, we won't know if it has anything to do with the experimental therapy that's being given or because of the changes that we're actually making. So the medications need to remain stable for many, many months. There's variations in the different protocols in terms of how long that is in order to give that as clean a signal as possible.
Hi, both of my kids have the SCN1A, exact same mutation inherited from their father. My son Kai, they're adults. My son Kai is very much on the Dravet end, and my daughter Sierra, who's 30, has normal intelligence. She's in college, getting ready to graduate soon. Do we have any hope that in the next 10 years, if she decides she would really like to have children, that she might be able to do that without passing this gene on?
You want me to take that one? So yeah, I actually don't think it's 10 years. I mean, there are a lot of ways to do pre-genetic determination, in terms of in vitro fertilization, and make sure that embryos are actually selected, because there's a 50 to make sure that actually doesn't occur. So it's not necessarily future. So yes.
We have another one online. Okay, let's try that again. There we go. Another one from online said, what kind of improvement can we see that will help us to understand that this therapy is working?
I couldn't hear it. Actually, say it again.
So what kind of improvement can we see to say that we understand this therapy is working?
That's a tough one. So, I mean, I know that most of these trials still use seizures as the primary endpoint, right? Because seizures are, I don't want to say easy to count, but it's at least measurable and objective, right? And so if we have a baseline of 4 seizures in a month, and then you get any one of these therapies, and over the next 6 months, you only have 4 seizures total, without any changes to the background medications, that's, you know, as good a proof as we are going to have that it was the intervention that actually caused that. Now, the developmental stuff, I think, is a little bit more challenging because development, even in a normally developing infant, takes time, right? And so even when we intervene with something that may be getting at the underlying cause, it may take 12 months for us to actually see the separation between what we would have expected to happen in the absence of a gene therapy versus what positive impact could be happening when the gene is improved. So I think it's still going to be focusing on seizures.